So in this part 2 of module 3, we are going to be talking about the role of triglycerides, the different kinds of biochemical functions, and then we are going to look at some other types of lipids. So if you see here, the triglycerides, what are they used as? They're used as a source of energy. They contain a lot of energy. They are also a little bit like they hate water, so they can be used for waterproofing because they're hydrophobic. And then the insulation part, like they are slow conductors of heat and good insulators to retain heat. So they are good for insulation. And that's why, if you see some of those animals that live in the polar regions, they're like really having a good fat layer so as to save them from all the cold. Protection, they also protect the internal organs from being, I guess, affected by any kind of physical pressure, trauma, whatever you call that. And so one of the examples of the presence of, fat storage in human body is in adipocytes. So if you see here, the white fat cells, there is a lot of lipids in there. So talking about triglycerides or there are different, you will see the different names for that, but it means essentially the same thing. 3 fatty acid with a glycerol. Okay, and so these are just the examples that we have talked about in just previously about oils and fats. Now, carbohydrates and lipids, the role of both of these is storage of energy, but one of them is short-term and quick storage, and the other one is longer storage. And you can see some of these differences. Take your time and read them carefully. I want to focus with you on the parts that would be a little bit complex. So for example, these triglycerides can undergo reactions, which is one of the objectives in this course to learn, is what kind of reactions they can undergo. So let's take a look. Please don't see the right-hand side. Let's focus on the left-hand side. It's the reaction called hydrolysis, in which the ester bonds are split by water so that glycerol is separated, and this is separated, which is the fatty acids. I'm not saying this is similar, but do you remember the assignment where you said galactose plus water, and then you said, what was it? What are the two things that is made up of? Let's see if you guys remember.
Glucose
And
maltose. No, glucose, maltose.
Galactose. Remember, I told you guys how to remember this. I said, glucose plus, I'm sorry. There's a mistake here. Definitely. I'm sorry. This is lactose. I'm sorry.
Glucose. Okay.
You guys should correct me when I'm making a mistake like this. You should not wait. You shouldn't be like, oh, this is instructor. No, you should. If you know this is wrong, it's wrong. Or something that's incorrect is incorrect. Okay. So this will be making glucose. This and resulting in glucose and galactose. So by this reaction, what I am trying to show to you is, in a similar manner, the triglycerides can be broken up into a glycerol, which is right here, and 3 fatty acids. Okay, so that's what it is broken up into. So I hope everyone can see how in one of the last slides we started with, let me reverse back for a second, glycerol plus 3 fatty acids and make the triglyceride. And now with the hydrolysis reaction, we have been able to convert that back to glycerol and 3 fatty acids. Do you see that correlation, that it can be converted back? It's not an irreversible reaction, okay? So that's one thing. The other part is the hydrogenation. So is it okay? I want to make sure that we finish a substantial part in the next 30 minutes. So as soon as you feel like you have a specific question, immediately write it in the chat. And I promise we can deal with it. For sure, I'll let Simone and Julie know that these are the specific questions that people have, okay? So if we go back here, hydrogenation, whenever there is a double bond, you see this double bond? This double bond can be broken and we can put H in here. So which means that CH double bond CH, and you know that if this double bond goes away, then we need one extra H here, because then C has only 3 bonds, and then extra H here. So that's what is shown over here. So that means that hydrogenation reaction is like you are forcefully adding hydrogen and breaking up the double bond into a single bond and making it saturated fat. And that's something that is actually done. Okay, the last part of triglycerides, okay, not the module, just the last part of triglyceride is saponification reaction. And saponification reaction is the one in which it's reacting with an strong base, okay? And it's reacting with strong base and it is forming a soap, which is sodium atom plus the fatty acid part, okay? So that is what it has released the glycerol, and this is what is making the soap, like literally the soap that you guys that you use, okay? So if you're talking about bar soap, that is what is made by sodium hydroxide. It produces hard soap, and potassium hydroxide produces a soft soap, such as shaving creams and liquid soaps. Okay, so probably that's more commonly used. And many of them are having this combination of hydrophobic and hydrophilic hair. And that's how they are able to remove the dirt, because they circle around the dirt, they kind of wash along with the water. So these 3 reactions are important. I wouldn't say super important, but I would say they're important. Okay, moving on to waxes. Waxes are esters of saturated fatty acid. Okay, so here, let me circle the important words. The word here is saturated fatty acid, and the word here is long chain alcohols. Okay, so you have to be very, very patient when learning this module, just because there's lots of terminology, lots of different things. So you got to tell yourself, hey, it's just going to be 30 more minutes, so I'm going to really, really focus. Okay, so each of these concepts are important. So what are waxes? They are esters of fatty acid and long chain alcohols, each containing 14 to 30 carbon atoms. Okay, so what is their role? The waxy coating on the plants, like you must have seen, there is a natural waxy coating too, and that prevents the loss of water. And there's one more slide on wax, I think. So I use the word here, esters of saturated fatty acid, so which means that these do not have any double bonds. So these are some of the examples of waxes. You see here, this is a beeswax or a carnauba wax, a jojoba wax. These are different kinds of waxes that are used for different purposes, which are written on the right side. So what are the roles of waxes? There are lots of applications in pharmaceutical and cosmetic industry. They're also used for the manufacturing of lotions and ointments, which are, of course, now we know why they're all used a lot, because when you're putting them on, then the body doesn't lose also so much water, because it's not like letting the water just evaporate. Okay, structural part of the lipids. Let's try to do this. Structural part, we are going to, I do want to tell you the signals, cofactors, and pigments is very small. The structure is the one that involves maximum terminology. So let's really try to do this to our best, okay? So the structural lipids are used in membranes, okay? So what are those lipids? So what are they used for? And, we are just going to start with looking at the classes. So major classes is phospholipids, there is glycolipids, and then there is this archibacterial ethyl So we are not going to be focusing on this. We're just talking about these two. This is not at all complex. Okay. Why? Let's just first start by talking about these terms. What is phospholipids? So that's a kind of lipid, as you can see, with the word phospho in it. And the glyco is the term with the word glyco in it. Okay, so what does phospho mean? Phospho mean that somewhere you have a phosphorus group. That's it. That's what it means. And what does glyco mean? Some kind of a carbohydrate group. Glycogen, glyco. Glyco is always about carbohydrates. Okay, so here you go. You have these two. So membrane lipids can be of two types, ones that have phosphorus in them and the other that have carbohydrate kind of structures in them. And then there is a third type. We'll come to that. Let's just focus on these. Now for a minute, let me erase this for you so that you guys stop worrying about these and just look on this side. Phospholipids can be of two types. Type 1, glycerophospholipids. Now, you know, you guys are smart. So you need to use that smartness to see there is glycero, there is phospho, and there's lipids. There's nothing to be scared, guys. We know that glycerol is for glycerol, phospho is for phosphate, and lipids is for those fatty acids, right? So let's find this. So I'm going to use arrow and try to show you. So glycerol, phospho, phosphate group, lipids, that fatty acid and alcohol, that's the lipid part. Glycerophospholipids. Oh gosh, we have a new term, sphingolipids. What is sphingolipids? So in sphingolipids, instead of having glycerol, we have sphingosine. Sphingosine. So sphingo goes with this, and the rest of this is the lipid. It does have a phosphate, but this one doesn't have sphingosine. So that is what we are going to talk about next. So let me clear this for you and go to the next slide, where we are going to talk about cell membranes. We're going to talk about these three, that the cell membranes establish this hydrophobic barriers, and these barriers will help select what goes inside cell and what goes outside the cell. So we are talking about phospholipid. Phospholipid contain an ester linkage. I'm right here, reading right here, contains an ester link between a phosphoric acid and an alcohol. So this alcohol can be glycerol, and that's what makes it glycerophospholipids. And the alcohol can be also a sphingosine, which makes it sphingomyelin. Okay, so let's take a look at this. This is what we're talking about. We talked about this phosphoglycerides and we talked about sphingomyelin. Okay, and what about glycolipids? So glycolipids are derived from sphingosine, but they have a carbohydrate group. So I want you to pay attention to this particular part where glycolipids is nothing but this part. Okay. Any questions on particularly on phospholipid, this slide, and any questions particularly on phosphoglycerides versus sphingomyelin versus glycolipids? And take a careful look at these units, you know, these structures. These structures are going to be your key to remembering this. I think one question that people often ask me is like, would you expect us to draw the structure in the exam? No, that's not the whole point. That's not why we are learning this. I will obviously, you probably know by now, my goal is not to make you memorize a lot of things, but make you learn a lot more than memorize a lot more. Yes. Oh gosh, that's excellent point. Yes, glycerol and glyco is completely different. Thank you for asking that. I wouldn't have thought of clarifying that. Glycerol is simply, glycerol is an all. It's a type of alcohol. So let's just go back 1 second. This is a glycerol. I'm just going to show you. This is a glycerol. See this? CH2CH, CH2 with a 3OH. That's a glycerol. Whereas glyco is always standing for some kind of a carbohydrate. So I'm just going back here. Great question. Very nice question. So I'm going back here. Yes, glycerol is for having that glycerol molecule. And yes, people can easily be confused with that. And let's look at a bigger structure in here, like just the same thing elaborated the entirely on the screen. Phosphoglycerides have phosphate molecule. They have this glycerol. which is CH2, That's what is present here, and that's the fatty acid. It's not shown here, but this R is long chain. It goes like really, really, really far. Okay. So that's what is this phosphoglyceride. In contrast, what is sphingomyelin? Now look at this. Now let's compare. Top is same, middle is same, bottom is same, a little bit to some extent. What is different? Okay, I think we found the difference. Hey, this glycerol thingy is not there. This is a completely different thing. This is this completely different beast, which is going all the way from here, like that, like that, like that, like that, like that. And that is what is sphingosine. You see this L-shaped thingy? That's the sphingosine. So sphingosine with the rest of fatty acid, phosphate, and a molecule is sphingomyelin. And then what is a glycolipid? Guys, that's your favorite. You see that? That's a glucose molecule. Or not glucose, maybe one of those similar to glucose molecule. And that's a glycolipid. It has everything from alcohol and fatty acid, but also have a carbohydrate attached to it. This is like extremely important. A lot of times, these glycolipids play an important role as a recognition, as a point of recognition for cells to recognize and do something. So we're going to talk a little bit about it. So now that we have somewhat come to terms that, okay, this is not too crazy, and I can, probably if I read it 3 more times, I probably will remember. So let's take a look at why do we need to even study this? Why are we like breaking our head trying to understand this? So glycerophospholipids or phosphoglycerides, as we mentioned, they are the ones that contain this fatty acid with the glycerol and a phosphate group. Why do we study them? We study them because they provide the basic membrane structure. So in each of your cells, do you know how many cells you guys have? Let's just like break this discussion for a minute so we can get back to. I'll give you numbers. Okay. So we're doing a poll. The poll is, do we have, okay, I'm going to write it in the chat. We have lots of cells, right? Okay, so how many cells does human body have? I'm going to give you an example. A, 1 million. B is 1 billion. Let's make it 100 billion. C is 80 trillion. How many cells does human body have? A, B, or C? Yes, C. You guys are smart. You guys are smart. Yes. So there is somewhere between 60 to 100 trillion cells. So why are we studying that? Each of your cell, imagine each of your cell has this structure. So why are you not going to learn about your own body and your own cells? So that's what the glycerophospholipids are. So these are the membrane lipids. They're fatty acids. And then there is this part, which is the polar part. Now look at this part, this box. This box is tricky. It's like, this is a little bit fancy because if you put, you can have a choline here. If you put this one here, that's ethanolamine. If you put that one, that's a serine, or makes a phosphatidylserine. That's a glycerol. You can put phosphatidylglycerol, or you can put this. So these are all the different boxes that can give it different properties. Okay, and your cells do need it. Why are we studying sphingomyelin? That seems like such a funny name, because sphingomyelin is the main component of myelin sheath of your nerve cells. Okay, so your nerve cells, when they're relaying signals, they need to be protected. So they are protected by myelin sheath. If they're not protected, they will lose signal and that will not be good for transferring the signal. So what do sphingomyelin, what is the definition? And that sphingomyelin contain phosphocholine or phosphoethanolamine as their polar head group, and therefore they are classified along with glycerophospholipids. Okay, so they do not have glycerol, they have sphingosine. Okay, so what happens if the myelin sheath is not working. So have you heard of multiple sclerosis? So in multiple sclerosis, what happens is that the myelin sheath is there, but the immune system attacks the myelin sheath, and that results in inflammation around the nerve cells. And so that results in multiple sclerosis. Also, it doesn't, then the signal cannot go from brain to the different parts of the body. And you can imagine that how that can restrict movement and damage the signals that go from brain to different parts. So what is glycolipid? Now, glyco, not to be confused with glycerol or anything. They are carbohydrates, they're sugar residues, okay? And they maintain an important role in the cell, okay? So I do want to tell you something super important, and that is the blood group. You probably know that you might know what is your blood type. I don't know if you know your blood types, but so the different blood types are because of different kinds of glycolipids. And so which means that this particular part, which is present in O or B or A, these are slightly different. And that difference on the membrane of the red blood cells causes these red blood cells to be of different types. This is AB, this is B, this is A, and this is type O. So very, very important. Okay, so what about steroids? So steroids are another type of lipids. What is difference? So steroids are the reason, what gives me reason to remember steroids is, there are all these ring structures. So there's like cyclohexane ring. Cyclo is the cyclic and hexane is 6 members. So 1, 2, 3, 4, 5, 6. So you can say cyclohexane and cyclopentane ring, 1 cyclopentane, 3 cyclohexane ring. And this is what, along with the side chain, makes it, or the R group, makes it the steroid. And you can see there are different types of steroid. We talked about cholesterol, which is just, hydrogen in here, but then it could be sitosterol, there's stigmasterol, there is campisterol, there is avenosterol, and so on and so forth. There's just a few examples of what can go in place of R. So what are their roles? So they have extremely important role. If the cholesterol is not present, actually membrane will get very rigid because all these lipids will come together. So they provide the flexibility to the membrane. So many hormones, that is estradiol, testosterone, vitamin D, they are all derived from steroid alcohols. Okay. So these are the different types of, different types of structures that we talked about in the membrane. Now let us see how they fit in the membrane. So if you see here, you will find here that in this membrane, you can see there is phospholipids. Now that we are familiar with this, there is glycolipid, which is these things protruding out. That's actually the hexanes. And then there is cholesterol, which is embedded in between. Then there is some other proteins and side chains. So this is the structural unit. There are also other proteins which are important. Like, for example, I'll give you an example. Have you heard of ACE2 receptor? ACE. Have you heard of ACE receptor? And if you have heard, in what context have you heard it? It's very popular these days. I'm going to write it down here. ACE2. Come on, I think you guys might have heard it. If you have not heard, then you need to be watching the news more carefully. Why do people talk about ACE2 receptors a lot? Anyone knows? Okay. So yes, thank you so much. Yes, someone knows. Yes. ACE2 receptor is where the coronavirus binds and enters the human body. Okay. So definitely that ACE2 receptor is where the COVID enters. So that's important. So those are those proteins which are transmembrane protein. That's where the ACE2 receptor is, where the virus binds. So this entire membrane structure is so important. This integrity of the membrane structure, flexibility, all of these different things are important. Okay, so this kind of model, and you might have heard this before, especially in Bio 101 or 102, is that it's a fluid mosaic model. There's membrane proteins and lipids that can rapidly diffuse or laterally rotate within the bilayer. Okay, so the membranes can be about 25 to 50% are lipids and 50 to 75% are proteins. Okay. So these are the examples of lipids that we already learned about, and then what the composition of biological membranes can vary in different animals, different species, different cell types. There are small non-polar molecules can cross by diffusion, but not everything can cross without any. Just like that, it cannot cross. There has to be a reason. There has to be a gradient, an active or a passive transportation. So just thinking about how all of this ties together in membranes. So cell membranes are composed of fluid-like phospholipid layer. Phospholipid bilayer. And this is what is shown here. These are the proteins that, sometimes molecule can just pass because of the gradient, and that is a passive transport. And then sometimes they need to be pushed against the gradient. Like, maybe there is a lot of, sodium on one side, but it has to be still be pushed out of the cell in order to maintain a fixed amount of sodium and potassium across the cell. So the reason why we are looking at this is to just say that the lipid bilayer plays an important role in ensuring that the transportation is occurring only through specified channels and things cannot just pass through the lipid. Pretty incredible. Last part, signals, cofactors, and pigments. Okay, so signals, cofactors, and pigments are important because these play an active role as metabolites. They play an important role in various reactions, and they are responsible for even colors, or just, act as an important cofactor when a certain reaction is happening. So lipids as cofactor in signaling molecules. So some types of lipids, they are needed in really, really small quantities, but they're extremely important. For example, there is prostaglandin, thromboxane. All of these are different types of, molecules, which are actually lipids if you see them. And they are hormones. Okay. So they serve as very important molecules which can change gene expression. It can make some protein, a lot of protein can be produced if a certain hormone is present. So it's extremely important because it can change, even if it is in small quantities, it can change the amount of proteins produced in certain cells in human body or other organisms. Lipids as vitamins. Yes, lipids do play an important role as fat-soluble vitamins. But vitamin D, A, E, and K are fat-soluble compounds, and they're made up of these isoprene units. And if you see on the right-hand side on this white block is the structure. So vitamin D is a precursor to a hormone that regulates calcium metabolism. And we're going to study a lot more about hormones in thinking the last module probably. Vitamin A is important for furnishing the visual pigment, and E is the important for protection of membrane lipids from oxidation. Vitamin K plays an important role in blood clotting. Why are we talking about vitamins now? Lipids form these fat-soluble vitamins, and they play an important role. I think we are almost to the last slide, if I'm right. Yes. So there are also other lipids such as ubiquinons and plastoquinons, and they are electron carriers in mitochondria. So they have not only just role as insulators and membrane protecting membrane or having that membrane structure or as sterols, but they're also serving as important, cofactor in terms of transferring the electrons. Then there are other roles such as for them to be active in membranes. So dolichols activate and anchor sugars to cellular membranes. That's important because we want to have those sugars act as important antigens. The sugar groups are then used in synthesis of complex carbohydrates, glycolipids, and glycoproteins. Whenever you see the word glyco, your eyes should light up. It should be like, I know, whenever you said glyco, it means something carbohydrate, okay? Whether it is glycolipids or glycoproteins, it's carbohydrate attached to lipids or proteins. Lipids, sorry for the typo, lipids conjugated dienes. They serve as pigments in flowers and fruit. So you see these colors. Have you seen probably seen the red cardinal or the goldfinch, and they have these colors, and these beautiful colors are result of these different kinds of lipids. They are also used for their medicinal value, and you can see one of the structures which shows here one of the lipids, which is not recognizable like a fatty acid and such, but it is another kind of lipid. It's a polyketide. It does not fall in a carbohydrate group. So that's pretty much the module. Let me just stop sharing and see if you guys have any questions for me. Any questions? I would encourage you to write down the questions, even if you did not understand something. So Simone and very nice question. I think I would say that's probably. So do you mean like skin? Did you mean like skin pigmentation? I think it's not just that. I don't think it's just that. I mean, so the skin pigmentation color may not be decided only by just that one factor. But it is a good question because, we are definitely talking about melanin, the production of melanin in there. And trying to remember, is melanin a lipid? Because that's going to be the one that's deciding our skin pigmentation. I can just look it up real quick. Is melanin? Try to see here. It says it's part lipid and part protein. Yeah, so it's not exactly lipid, but yeah, there is some part of it. So that just means it's a little more complex than that. Any questions you have, like let's say you did not understand nomenclature, write down, still have questions in nomenclature, triglyceride, didn't understand the sphingomyelin part, whatever it is, write it down.